It's a pleasure to be here, and I appreciate the opportunity to come interact with this group and the invitation to give everybody a lecture on genetics at 8:15 in the morning. No, it's not going to be that way. But I do have some things I want to talk about and some preliminary evidence and some things that we've been doing, some information and some data in the genetics world from some other species that might hold some relevance to us. And so this is not necessarily the order that the talk is going in this morning, but I do have these points kind of interwoven. And when we think about intensive cow-calf production systems, they do offer some special opportunities and some advantages potentially on the genetic side of things in regard to data collection and thinking about that stuff. So I am going to talk a little bit about some basic aspects of genetics, and then we'll move into some more interesting data and discussion. But I wanted to start with this slide, and I made this, I guess, last year after visiting some different places. And so we're used to seeing the beef carcass on the left there. Typical of our production system in the United States is the steer carcass, somewhere around 4 inches, 0.4 inches of 12th rib fat thickness after being in the feed yard for roughly 6 months, 18 months of age, and something with under 25% Boscinicus influence. Contrast that to the bull carcass that's 30 months old over on the right from a grass-finished operation that's 100% Boscinicus in a Latin American country. Now, those are not the same products by any means, are they? And so if you produce one in the production setting of the other one or attempt to produce one in the production setting geared to the other one, then that's not very effective. But within both of those production contexts, there's the ability to identify animals that could have contributions from the genetic side of things, but we would not make the same recommendations in all settings. And so I think this is an obvious case that maybe sets the tone for that. And if you think about some things that we do on the intensive side of things, a lot of the world looks at that and tries to replicate that with some success in many cases, but with limited success in some others. And so I've got this table in the paper in the proceedings, and how intensive do we want to go really depends on is it cost effective for us, and do we push the limits of biology more than they need to be. And we have to think about how all the different traits that are involved in a production system kind of fit together. And so there's not really any easy or simple answers, but if you think about on the left side here are just some bullet points that I thought if you think about really trying to push the envelope on some things, a lot of this is what a lot of our producers are doing already. You know, we don't try to get heifers to calve at 18 months of age. But we do see that, right? And that's a screw up when that happens. But there are heifers that will start cycling early enough. And if we really wanted to make that a priority, at least in some breeds and some settings, then we might want to be able to push toward that. I'm not saying that's necessarily the best thing to do, but we can move some of these traits in directions if we want to. We're used to castrating all of our males that are not destined for breeding purposes. And that happens at various ages, but that doesn't happen in a lot of places, right? And so we have to know something about the genetic potential of the animals in the context of what we want them to do, and also how we want the whole system to evolve and change over time. And when we think about genetic management and that information, just knowing some of the genetic aspects at one point in time is really not the most effective use of genetic information. It's a long-term, multi-generation concept. Now, if you think about how our industry in this country really uses genetic improvement and some practices, the genetic change really relies on the seed stock sector, right? And so the most common product there is selling young bulls to commercial herds. And so these are 18-month-old, 24-month-old bulls. And in regard to genetic information, by nature, they're fairly low in accuracy on their EPDs. And so we've tried to incorporate some technologies and increase the accuracy of those with some of our genomic technologies. But that group is used to thinking about reproductive management and some of the reproductive assisted technologies a little more than some of our other sectors. And so most commercial cow-calf operations rely on buying those bulls from the seed stock herds and putting that on some type of crossbred cow base. And so the limited amount of information that is used there limits the amount of potential and the efficiency of some of that use. And so when we think about genetic change or genetic improvement, this is really what is the heart of it. And this is our historical way of thinking about genetic change. We've got a bell curve, we've got a continuous distribution for some trait, we know what the average is, and we're trying to shift that average down the number line. And so genetic improvement, in most cases, we think about getting more production or a bigger animal, or, it's usually shifting that to the right, but in some cases, we may want to go the other direction. And so the term for that is selection response. We know that it can happen. We measure that in different ways. Genetic trend is something that our breed associations talk about, and the change in average genetic merit, or EPD, over time. That's the same concept. We're not showing bell curves, but we're talking about changing the average of those bell curves over time. And this is data from the American Angus Association. And so this represents animals in their database by birth year from 1972 through 2014. And so some of you are used to seeing some of this kind of data. And so there's 3 lines on that graph. The solid dark line at the top is the average 365-day yearling weight of purebred Angus bulls in the database, and that goes with the number line that's over on the left here. And then for the heifers, the 365-day average weights over time are here with a solid gray line on the bottom, and the average EPD for yearling weight across those years for those animals born in those years is the dotted line. And so if you look at the change in EPDs, then that is, we could say, only genetics, right? But if we look at that change in yearling weights, even within a seed stock operation, that's not just genetics, right? Because our management practices and how we feed animals is part of that. And so the real usefulness of genetic information is in combination with other things, right? We don't try to solve things only by health management or only nutrition or only genetics, and the interactions of those things are really the key to trying to get at efficiency from a production standpoint and a whole system standpoint. So we had some discussion about this yesterday. We are all familiar with the concept of increasing carcass weights, and the numbers there are different a little bit depending upon the source there. But we know we continue and we can increase weights of cattle. And how big can we make them? I don't know if we know the answer to that. How big do we need to make them? I don't know if we know the answer to that either, but that trend has been linear and increasing for quite a while. And the bottom figure is from this year's Beef Improvement Federation meeting that shows the breed-adjusted average yearling weight EPDs across this same time span from the early 1970s till recently in our 7 major beef breeds in this country. And so we know that those change over time. And so for our stereotype of our British breeds being more moderate size and lower milking than some of our other breeds is just not true today. And there's a lot of variation there, but if we're talking about the average or we're talking about the whole distribution, then those are two different things. And so we know that we can change these traits, and a lot of our historical ways of doing this has been effective for some of those what I would call less complex type traits, and weight at different ages is not as complex as some of these other things that are really important to us. And so I took carcass weight and incidence of respiratory disease and percent calf crop weaned in some different data sets across the same time span. And like I said, if we look at different sources, some of the numbers here may be a little different, but we mentioned increase in carcass weight. So here's some data from the National Beef Quality Audits that were done in 91 and 2011 over that 20-year time span, and we know that those are increasing. If you look at things such as morbidity and mortality rates in feed yards, that hasn't improved during that same time span. And now, why might that be? There could be a lot of potential reasons, but that's a different kind of trait from a genetic standpoint too. And so that's a lot more complex phenotype than yearling weight or carcass weight. And depending upon what data set you look at, we know percent calf crop weaned, Dr. Rasby talked about this yesterday, is the key to a lot of things in regard to sustainability financially on cow-calf side, and depending upon what data set you look like at there's not a whole lot of increase or improvement in some things like percent calf crop wean. And so those kind of traits all have genetic components to them, but a lot of times they go undocumented, or there may be some other genetic mechanisms that are going on that may add to their complexity. And I want to spend a little bit of time going into that concept a little bit more. So I have to just make sure that we're on the same page when we're talking about some of these different terms, okay? And so when I say genome, you know, that's just the complete genetic makeup of an individual. That's the entire DNA sequence. And there's different forms of genes. We call those alleles. Where they reside on chromosomes are called a gene locus or loci. And when we think about genetic influences and effects, then both the effects of the individual alleles or the effects of those combinations of alleles, and we call those genotypes, can be either additive or non-additive. And so if we're trying to estimate the genetic merit for yearling weight, we don't know how many genes affect yearling weight, but theoretically there's those effects that add up across all those different genes. But we know not everything adds up, right? So the most common example that we might use in cattle on non-additive genetics is black versus red inheritance. We can have bulls that are carriers for red. That's because those two alleles do not act in an additive fashion. If we think about the total variability in performance, we can represent that with a bell curve, but we can also think about that as a pie graph. And so for every trait, there's a certain component or a certain amount of that trait that's due to additive genetics, non-additive genetics, and we say anything that's not attributed to genetics is environment, and management is environment. So environment is just used to represent something that's not genetics. And so when we see or hear somebody say, if we have a trait that 10% of the differences are explained by the individual animals' breeding values or EPDs or that kind of stuff, then the other 90% is environment. And that's not necessarily true, because the non-additive aspects are important. And so we're used to thinking about those, and you may have heard the term and be familiar with hybrid vigor or heterosis. Those are non-additive concepts. The level of variability and performance due to those things is a function of the breeding system, and it's not passed from parent to offspring like the individual genes are. The effect of the individual alleles or genotypes or the additive aspect, you can think about animals sort of being like bags of genes. The genes that are in there affect that performance, but some of those genes interact, and that's the non-additive stuff. So we've got potential variability for all these things. That's not a new concept. Anybody that's managed different sets of cattle, even if you didn't understand or think about genetics, recognize that concept of variability. And all of these components have their own levels of variability. So if we think about trying to relate some of this specifically to some intensive situations, for many years, excuse me, it's been recommended that breeding animals be selected in environments that are relatively similar to how their progeny are going to perform. And we have some challenges to that large scale across this country because of the design of how our industry is. So we have widespread use of artificial insemination, for instance, right? And so if we're using semen on bulls that are very desirable for some carcass trait, and they are really adapted to a different part of the country than what we're using them in, then we might want to think about that just a little bit. If it's a terminal situation and we're not keeping replacements, then it probably doesn't matter, right? But if we have cattle that come into a different area and we're retaining some of the genetics in our herd, then those animals may not be the best fit for the whole overall production system, even though for that one trait they really may move our bell curve a lot. The other thing is that we know that we have lots of environments where our cow herds are that's very different that's entirely different from the finishing phase of our industry. And so even if they're in the same geographical region where there's a feed yard, the conditions and management on that ranch are nowhere near the same environment as in that feed yard, right? Because of how we manage those animals and feed them. So from a whole systems perspective, both of those things are challenged if we don't try to connect all the dots. And our industry has looked at genetic improvement in a particular sector a lot more than trying to look at things from a whole production system standpoint. And so that's one of the reasons why I think this meeting and this symposium is really so good and effective, is that we have several different people talking about trying to connect the dots on a lot of different things. So in regard to intensive systems, this may not be complete nutritional control. It might be just some time of the year, so we might say targeted instead of complete. But that is a big advantage from a genetic standpoint, because we have things and we know what those environmental influences are. And we can account for that. And one of the biggest challenges on the seed stock side of things is this concept of contemporary groups. And so animals that have been, managed the same and under the same environmental conditions, we call those contemporary groups if we're turning in data to breed associations. Okay, we have contemporary groups that feed yard managers think about all the time, but we don't call them contemporary groups. We call them pens or lots. It's that same concept, right? So those groups of animals are not managed or thought about from a genetic standpoint, but we can. And so in these different intensive systems, we really have the ability to track animal performance across multiple, it's not an industry sector if it's ownership, but across multiple time points in regard to production of market animals, and also in development of breeding animals if the replacements are being produced in the herd. There's a lot of talk, and there's a lot of good discussion, and there's a lot of good applications of genomic information, and this continues to increase, and it will continue to be more and more cost effective in the future. For a lot of people right now on the commercial side of things, probably just knowing what bull sired what calf and the ability to use multiple sire breeding pastures and get that individual parentage information is really, really beneficial. We have a lot of tests that are on the market for single gene, what we would call Mendelian inherited traits, red versus black, horn polled, some diseases. But the incorporation of genomically enhanced EPDs is really going strong, and that will continue to be a focus. We have most of our breed associations either trying to implement that or seriously thinking about implementing that. And that alone may not answer all the questions that we might have. So we're all familiar with these different genome projects. Human Genome Project was the first one. And this graph just shows how much reduction in cost in getting the actual DNA information has occurred over the past 14 years. And so it used to cost billions of dollars to determine the entire DNA sequence of an individual in a particular species. And that technology has just evolved so much. It looks like, if you think about the cost on either total cost per genome or in how many dollars per million base pairs it is, it looks like in the past few years that's flatlined, but it's not. It's just because our scale is so big that you can't tell how much it's decreased. And so just from 2009, we said it cost something like about 78 cents to determine 1 million base pairs of DNA. And this year, that same amount of genetic information cost us about 5 cents or less to get that. Or if we wanted to genotype an entire individual, then that's $4,500. That's still a lot of money, right? But the way that the technology is going, there's going to be more and more genetic, molecular level information available to us. And a lot of people are thinking about how can we use the total DNA sequence in evaluations. And that's just almost mind-boggling to think about how much data that is. And the reason that that offers some advantages is, maybe you can see that there's some different colored dots up here. The more detailed coverage we have of the genome, the better we are to identify the genes that affect the traits. But maybe more importantly, we're better to identify what some of the different interactions might be or regulatory aspects. And so this is a picture from one of my colleagues, Dr. Claire Gill, over a 2 million base pair region of one particular chromosome. And so we've had the 50K SNP chips single nucleotide polymorphism. That just means markers where there's a single change in the DNA base from one letter to another. And so 50,000 genotypes on an animal is a tremendous amount of information, right? But there's almost 3 billion potential genotypes in an individual. So if we are using the 50K chip within regions, yeah, we see some hits related to some traits, but when we start to incorporate things that are called sequence scales, so we have animals that we've got total genome information on, and so when there's 14 million potential genetic markers, then you can fine tune and find anything that you want to look for. And so we've got more data by far than we have more knowledge in the genetics field right now. And so it's a challenge, but it's a good challenge to have. Now I want to spend just a little bit of time talking about some genetic results and philosophy in both the human and the rodent model species. We don't evaluate genetics in people the same way we do in livestock, obviously, and we don't evaluate it in livestock the same way we do in mice and rats and some of these other model species. But for several years, we've had a lot of genetic information in human medicine and things like that. And so we know about what the heritability ought to be for these traits. And so when they look at this concept of genome-wide association, so that's what this refers to. If you have all the genotypes across the whole genome and you have the phenotypes and what are the associations there, then that doesn't do a great job in explaining what the total genetic variability is. And there's been a lot of people speculate on why that might be, and some of those speculations are there at the bottom. The one thing that is evolving more is this concept of epigenetics, and we know that that is important for a lot of traits in human medicine. That's important in regard to a lot of cancers and other health aspects, and has been widely documented in mice and rats, and has started to be documented in cattle. And if we don't look for those influences, even if they're there, then that just adds noise into the production system. So how much of those type of effects really impact our production, we don't know. But I think we probably ought to investigate that a little bit more. So anyway, that's a challenge in the human side of things, and there's not a consensus on that. Now, this concept of epigenetics was brought up yesterday in some of our discussion. And so epigenetics is something where there is a pattern of inheritance, but it doesn't follow our traditional Mendelian inheritance patterns. And so the fellow that coined that term was a guy named Conrad Waddington that was studying fruit flies in the 1950s, and said that there were some heat treatment on these fly pupae, and that changed their phenotype for the rest of their life and in their descendants. And so that's not what we think about as being traditional inheritance, right? So we know that there's some traits that follow that in some of our livestock species, but how prevalent that concept is, we don't know. And why might that happen? A lot of that is related to this concept of adding methyl groups or methylation at the DNA sequence and dictating which genes get expressed to what degree. And so there's a lot of regulatory aspects that may be coming into play. And there's been research done in several lines of mice that show when we alter the diet for different aspects that can change the methylation patterns. And that's been reported in regard to color pattern and body composition and things like that. And so we have to think about things differently when we have animals that are genetically identical at the DNA level, but they don't have the same phenotype. And so this is a picture of two mice out of one study. This is out of Randy Jerdel's lab in North Carolina. And so those two mice are genetically identical because they're inbred strains, but they're out of different litters. Okay, so the dams are genetically identical, but they're not the same dams. They're not clones. They're established from many, many generations of brother-sister inbreeding. Okay, and so the diets of the dams of those two litters were altered to produce some different phenotypes and different color patterns, and not all the same ones within the same litter have the same color pattern. And so if that's going on, how do we incorporate that into some of our genetic evaluations or our production systems is a challenge. There's ways to do that, but some people don't want to think about that because it changes how we think about things. When I was in graduate school in the early 1990s, we had some people working in cattle cloning with nuclear transfer, and so at that point in time we couldn't clone an adult animal. And I remember talking to somebody that had a set of clone mates, and they had calves born that weighed anywhere from 72 pounds to 140 pounds. And they're supposed to be clone mates. They all came from the same fertilized egg, and they were just split. How is that possible if they're genetically identical? And so that was thought to be weird. But that same phenomenon was true with in vitro fertilization. We had large offspring syndrome in cattle and sheep associated with that. What's going on there? And so even when we clone adult animals with somatic cell cloning, we can get that. And so these are two cattle fetuses that are clone mates at day 80 of gestation, and it shows how much different the fetal development might be. And if we look at the development of the placentomes and the placenta, then those are also very different. And what's going on is potentially there's differences in these methylation patterns that happen for a variety of reasons. So in vitro fertilization, they modified some of the media that they were using and got a handle on that. But some of this occurs naturally, and we know if you think about the different environments that our pregnant cows are in at different stages of gestation or from one year to the next, potentially there could be some of this impacting several different production aspects. So the good news about the increased genetic information is it's going to allow us to ask some more questions and pursue these things to find them out. But implementation, if they're there, and making sense of them is a whole different deal. But it looks like the epigenetics thing is really something to be considered in both the human and the mice world. And so a lot of people have speculated that there's some genes that are inherited where the expression is different from the male versus the female line. So if you inherit it from the sire or the dam, there's a different level of expression. That's just called imprinting. So if you see that, or genomic imprinting, then that's what it's talking about. It may be occurring from methylation probably, but there may be some other things going on there too. So on the human side, they're interested in using some of this for a variety of different reasons, and some of the information that's just come out recently really makes me scratch my head a little bit, because in human field, it looks like these methylation patterns may change over the lifetime of an individual. And we've got some people that are interested in looking at forensics and trying to find a blood sample and then look at the methylation pattern of that and trying to get some idea about predicting the age of the person that came from. And so in several studies where they knew the people and the source of the blood, they were able to predict the age across a long time. And just this year, there was a report that came out where people were looking at methylation patterns regarding ages of men that their children were born, and it looks like there's some patterns that there might be increased autism rates relative to men as they get older. And so this concept that our own genetic makeup may change, or that the value of an animal as a parent may change over its lifetime, really adds a layer of complexity to management, right? So that doesn't mean we ought to just throw up our hands, but it does make things a lot more complicated. And this overall methylation rate in people looks like it might be related to a variety of health aspects. So I would just want to make sure that we're all on the same page when we say something that's inherited or expressed differently or imprinted, what we're talking about. And so if we had some gene that we knew affected weight, okay, so we've got two different genotypes here, and we've got a 40-pound difference, and the average of the heterozygotes was right in the middle, then we would say that that has an additive effect, right? When we add one copy of the A1, we get an extra 20 pounds. And if it's not following a traditional inheritance pattern, we might have something like this. So if it was imprinted and you had the A1 allele expressed only or expressed more when it was inherited from the sire, so that's what this genotype means. So we got the A1 from the sire and the A2 from the dam, or the A2 from the sire and the A1 from the dam. If we had some different levels of expression, the average of all the heterozygotes would still be 980, right? But there's maybe some extra information in some of that. And in the 1960s, there was a lot of research being done on making different crosses and studying hybrid vigor, and the F1s were made different ways, and all the F1 data were grouped together. Not all the F1s are the same, and the model for that in some of our Boscinicus Taurus crosses really don't fit that pattern. So this concept of imprinting is really not on most people's radar, and honestly, how much it needs to be is still left to be determined. But we do have some people investigating that, and so this was a report that came out in Journal of Animal Science a few years ago, and a large data set, and it was all pedigree analysis, different production system. This is from EU, German, Simmental, and they investigated some of these carcass traits and how much influence there was relative to things that were inherited from the sire line or the maternal line. And they said of the 25 traits they evaluated, there were 10 of them that showed some of these significant, they called it imprinting. They didn't have the actual DNA data. They just looked at the inheritance patterns. And so there were some traits, and I don't know what all these are exactly, but FAT score had almost an entirely maternal control, meaning that the genes you inherited from the father and the inheritance there didn't seem to matter, and explain the variation of that. And in regard to some of the individual muscle weights or things like that, some of those were right the opposite. And so it could be that we have a lot of things that are sort of canceling each other out that are not recognized, but that's one study, and whether or not that can be repeated, we don't know. It goes along with some other things though. Now, what I want to spend a little bit of time and wrap up on is some data that we're really familiar with that may relate to some of these patterns. And so it's been known for a long time when we cross Boscinicus and Bos taurus that we get widely different birth weights of those calves. And so if we have Brahman bulls bred to Angus cows or Hereford cows, then that type of F1 has a really high birth weight, and the difference between the sexes are really, really exaggerated. Okay, so it doesn't really matter what kind of Boscinicus and Bos taurus cross we're talking about. That has been documented in several places in the world with several breeds, and we've known about that for close to 50 years. But it's always been thought that that was just a maternal effect. And so this is some data out of our research center at McGregor over some time, and these are Hereford, Brahman, and F1s. Okay, so the sire and the dam are listed here in the order first. And so purebred Hereford calves, this is what their birth weights are, males and females. Purebred Brahman calves, okay, Hereford sired F1s and Brahman sired F1s. And you can see how much difference the average birth weight is there on the Brahman sired F1s, and how much different the growth rate is, or the birth weights of those sexes are. And then Angus and Allure, same type of thing. Animals that were produced in contemporary environments. And so that was kind of interesting, but nobody really knew what to do about that. But that's also been documented in embryo transfer calves that have all been put in the same type of recip cow. And so this shows a couple of graphs from a paper a few years ago in making Angus back crosses. And so Angus bulls bred to F1 cows, and F1 bulls bred to Angus cows, and then the Boscinicus back crosses. So Boscinicus bull on an F1 cow, F1 bull on a Boscinicus cow. And that same pattern is there if we're talking about purebreds or different fractions of breeds. And so a lot of people speculate there may be some non-traditional inheritance or imprinting or something going on. And we recently went back and studied some historical data from Grenada in the 1980s, where they had a lot of different crosses in Brahman and Simmental. And it looks like there may be some interactions going on between things that are inherited on the X chromosome and some other places. And that also matches up with some stuff that's been found in mice when they made some different species crosses. And so looking at the information in the human and some of these other species gives us some insights on some things to look for. And when we make some other crosses besides the F1s, we also think we're seeing some unusual aspects. Okay, and so these are F2s. So when we breed F1 parents to each other, that's just the second generation. We call those animals F2s, right? And so these are Brahman Hereford, all half-bloods, but the way that this is set up, this means, okay, these calves were out of an F1 bull that was Brahman sired and an F1 cow that was Brahman sired. Or these were out of an F1 bull that was Hereford sired and an F1 cow that was also Hereford sired. Y'all follow the letters there? And so again, on the birth weights in some of these crosses, some of them are different than others. So these are the birth weights, and then the difference here between the male and the females maybe gives us some insights into some different genes that might be expressed differently. And so in some of these crosses, not just an average difference, but there were some really exaggerated difference between the males, and in some cases the females were heavier, and for pre-weaning gain from birth to weaning, the female calves had more growth than the males in some cases. And those are just simple means, and that's a small data set. So that would be considered preliminary data, I think, but that really kind of makes us scratch our head. And then this table's got a lot of numbers in it. I apologize for that, but I didn't know what to cut out. This one's in your proceedings. We've made contemporary F2 and F3 Angus Nelore calves out of our genomics project for several years. And so this is only the males. We've evaluated the males after weaning for a variety of different things. So this is set up the same way, but now A&N or Angus and Nelore. So we've got the 4 different types of F2s there, and then we've got F3 animals. And so there's some differences. When we have the same sires produced in 2 different types of F1 calves, sometimes there's a difference, right? So this is not differences in the individual animals, but in how they were made. And so if you look at birth weight, right, some of those are heavier than others. But what surprised me when we started looking at this is that there were some differences later in life in regard to when those animals were 15 months old and as carcasses, and there were some differences in marbling scores. And the patterns at those later points in life were not the same as the patterns that we saw early in life. And so this goes back to the way our industry is structured in genetic strategies, is that we know what goes on at the cow-calf operation, and somebody knows what happens at the feed yard, and somebody knows what happens at the packing plant, but a lot of times we don't connect all those in a total production system. In intensive systems, there's potential to do that more readily, at least for parts of that. These are the same numbers for 3 of those traits off that previous table, but I thought it'd be easier to see the bar graphs and look at all those different numbers, right? So for birth weight, we've got, these are out of Angus sired F1 bulls that were a lot smaller than the F2s that were out of the Nelore sired F1 bulls. For weaning weights, there were some big differences, and probably that's a function of we've got to remember the F2 calves are out of F1 cows, which we know are extremely productive, and for yearling weight, a different pattern. And a similar pattern to this for the marbling scores. And there were no differences in fat thickness, but there were differences in intramuscular fat. So my goal here is not to have anybody think these are the kind of cattle to produce. This is a model to study some things like what's been done in humans and mice. And so if the question there at the bottom, if this effect is real, we still haven't figured that out yet, but it looks like it might be, and it occurs in other types of cattle, then that potentially could be why we've got some low heritability for some of these traits, or why it's been so hard to uncover some genetic influences for some of our more complex traits. And so some of this may be going on in different breeds or different crosses, but just at a smaller percent and sort of under the radar. Okay, so somebody sent me this picture a few years ago. I love this picture. And this is human nature, right? We want easy answers, and sometimes we don't realize how hard the question is that we're asking. And so when we've got a bunch of phenotypes and we've got a bunch of genotypes, it's easy to think if we just have the phenotype and we have the DNA, then we can find the genetic markers and incorporate that in and know all of our genetic influences. And that may not be the case, because we know that these different traits that are of economic importance have a different, what we call a genetic architecture. It's not the same degree of genes or genetic influences that can be going on. And so I like to use this example. So think about some different traits that are important in the cow herd, and there's a bunch of them, right? And some have more direct economic values than others, but a trait that's not real complex to me is something like foot or leg conformation. So, you know, there's not a whole lot of guesswork into assigning the phenotype or the performance for that trait. How much feed we supplemented in the wintertime is probably not going to really affect that trait too much. And so that kind of trait has a higher heritability because the genetic influences are not that complex relative to predicting the phenotype. And on the other end of the scale, we've got something like if we had average annual net return per cow. So do you think there's any genetic influences on that type of trait? Yes, but for a bunch of different reasons, right? Because there can be differences in breeds or family lines, or differences in regard to hybrid vigor for a bunch of different traits, right? And so that's a highly complex trait. If we did a genome-wide association on that, we'd never get consistent results, because we might get a genetic peak on something that this year it looks like there's a genetic influence going on, but next year, due to market prices, that genetic effect on that trait is not the same, right? And I don't think that's the case on some of these less complex traits. And so we may need to investigate some of these more so that we can characterize them really well and predict some of the more complex things, instead of just doing a straight analysis on some of those. And I think health is a lot like that, right? Is it likely that we had two animals that were sick or healthy, but it occurred for different genetic and management reasons? Absolutely. And so a lot of these traits that are classified just one way or the other adds to their complexity. So I've got just a couple of concluding slides here to wrap things up. We know that some of these non-typical, non-traditional genetic effects are important in other species. If we look at some of the work that's been done at Nebraska and Wyoming and South Dakota State, North Dakota State, we know that in regard to fetal programming, there's potential to change how some genes are being expressed there due to maternal nutrition. So there's growing evidence that that's important in cattle. And the people that are involved on the intensive production systems on the cow-calf side, I think, are really structured to maybe investigate some of these things, or at least document some of these things to a more degree, because you know what the environment is, and it's a more repeatable environment from one year to the next, even though your commodities may be a little different, right? You're formulating rations for a certain level of performance. And that helps take out some of the noise to identify animals that are different genetically. And the one thing that our industry, and in particular intensive systems, can do is keep track of individual animal data if they want to, because those animals are evaluated on a regular basis. And we know if we have the ability to keep track of individual records, if we want to make genetic change, we can. We're better positioned to do that. On the business side of things, I didn't talk about this at all, but you know, some of the larger intensive systems may actually have a service to provide for some of the seed stock operations as a progeny test site. That's if you're wanting to produce your own cattle versus somebody else's, or get semen donated, or charge people for doing things. I think there's some potential to do that. If you're wanting to produce your own replacements, then that might not be a route to go. And then the last slide here to think about is, if we're talking about genetic improvement, we've got to have accurate phenotypes, we've got to know something about the contemporary groups, and we've got to know the parent information, and we've got to think about things for more than one generation. That's what genetic improvement is. So if we have those things, and the build for whatever we want to measure, then we can try to parcel out what some of those different influences are. And there's a lot of people that want really, quick, simple answers to these genetic questions, and they're really a lot more complex questions than what they're thinking. If you're just trying to use some DNA information to sort calves, in a feed yard, and that's all you know about those animals genetically, that's not the most effective way of doing things. If that's all you can do, then that's all you can do. Okay? Thank you very much for your time. I'm going to stop right there, and if we have time for questions, or if anybody wants to run me out of the room, we can do that. Thank you very much.
We got time for questions, so fire away. We got a microphone in the back somewhere? There we go.
Yes, my question would be, has there been much research done where you're looking at, let's say, continentals rather than Bos indicus and English on the sire female side to see if these same type of presentations occur?
There has been some of that. Most of the work that's been done where they made the different crosses either lump the different types of animals together, and so they didn't look for that. There has been a little bit of evidence from, I guess, 2 papers I can think of that they saw some potential for that. It's just not been very widely looked at.
On your German Simmental slide, it showed that the fat deposition was very much maternal. Would that indicate that the maternal side has more to do with the grading than with the marbling?
Well, so that particular, bring that back up. That, my understanding of what that is an external fat score. Just because in the EU they don't have a measure of marbling, and so I think that's just strictly external fat. Now you hear people say in different cattle breeds that there's certain family lines that tend to make better sons or better daughters, or there may be ones that have the ability to have different fat deposition patterns. Based on this, we don't know that. That's just the external fat. Do you have any thoughts on how that might work? Or what are you speculating?
I was thinking on the grading we get on the Jersey Gelby cross heifers, that they're very high grading. And I thought maybe that was the maternal side because the Jerseys are #1.
So that brings up a really good point. When we think about fetal programming and maternal influences, for most of our production systems, the maternal effect and the genetics are kind of all wrapped up together. And so it's kind of hard to say how much of that's due to the actual genotype of the calf versus the type of the cow that raised the calf, because those are, I think there's potential for that, but we haven't gotten the data to break all those things apart.
Dr. Harry, I'm curious what your thoughts are about, let's say you mentioned progeny testing and utilizing the more intensified systems for that purpose. What are your thoughts about ranking, let's just say cows, simple traits like fertility and weaning weight, I guess, in that environment? And can we use that ranking for an extensive purpose of production system?
Okay, so if you evaluate the animals and their level of performance under one production system, how that translates to another production system, that's a great question, Dr. Lallman, and I don't know the answer to that. We need to know that, and I think if we have some of the same bloodlines that are being evaluated in some of these intensive systems that are also being widely used in the industry, then we can probably help answer those questions. My personal thought is that there's some degree of relationship, but it's not the same, and so how much is it different? I don't know, but you know. Cow longevity in a controlled environment versus in a range situation is not the same thing, and it doesn't have the same value. And we shouldn't try to make it the same thing necessarily unless we're trying to use the same genetics for everything. And if we're trying to use the same genetics for all aspects, then the average animal is the best. But the average animal is not the most precise match for a particular environment. Does that make sense? That's.
So I think this is related, but looking at your data toward a little farther along, that was in the lower Yeah, this stuff here.
Uh-huh.
So did you also keep the data within the year and keep weather data, keep any kind of feed differences? And did you quantify that out, separate it out to look for differences year to year?
Yes, and so these are contemporary animals, and so these cattle were produced together. This data set is across 4 different years, and so after, from the yearling weight on, they were fed together in the same diet, and that diet was consistent across years. And then to get the carcass weights, they were all shipped to a commercial feedlot and finished there at the same feedlot in all 4 years. And so when we go to do the statistical analysis, there's a year effect there. But in this case, there wasn't a year interaction with any of those other types. And so it's been consistent that all those have been evaluated in the same general environment.
So Andy, I'll ask the question that you alluded to, but nobody has asked yet. So.
Why am I still here?
No, sir. I won't ask that because we have to ride home together. Your picture of the fallen down house, and so our very human desire to reduce complexity to simplicity. So what is your perceived value for genome-wide association for highly complex traits for which the phenotypes are poorly defined?
Yeah, that is a great question. If you have animals that are healthy or not, and that's your classification, is all healthy equal, or is all unhealthy or sick equal? And the answer to that's no. And so if you compound that across time and not know what all the different inputs are, I think that that's probably not the most effective way to analyze that data. Now, that doesn't mean I've got a good answer for that, but if you had more information on some of the component traits that allude to that, then I think that's got more potential on looking at some of the genomic information and describing some of those less complex traits, and maybe looking at some kind of index function or something, and that also incorporates the genetic information with the nutritional, environmental information to predict a phenotype. And so instead of analyzing the more complex phenotypes, maybe we ought to analyze the less complex stuff and try to predict some of those more complex things. And again, that's a lot easier said than done.
One more question?
Yeah. When you were talking epigenetics, did you say that recip cows had could have a measurable difference on the animal produced? And if so, was it really noticeable?
Well, I don't know if I said that or not. There is potential for that. I mean, some of the cattle that we've produced have been by embryo transfer. And so it depends on what trait you're thinking about, and it also depends on the level of variability in the recip cow that you have and how different she is from the type of calf that she's producing. There absolutely is a way that the recip cow could have some impact on some of those traits like birth weight, gestation length, weaning weight from milk production. But we think about that just being a temporary environmental effect that goes away. So, but if the gestational influence of that recip may impact the expression of some of the genes in the calf that she's carrying that doesn't have any impaired genetics, that potential is there. We haven't looked at that in a great degree, but there may be some of that. The statement I made about the recips is that if you took those different genotypes of calves and then put them all in the same type of recip, you still see some of those big differences. And so it's the effect of those reciprocal effects were not just due to the type of the cow that the calves were being gestated in. but what you're saying has potential.
Andy, yesterday we had discussion about maintenance energy requirements and the potential for semi-confinement operations to reduce maintenance requirements. You know, the number yesterday was maybe up to 30–40% because we're controlling the environment, lower activity, we're feeding high-grain diets at a limited level.
Mm-hmm.
So what is the potential to be able to reduce maintenance requirements from a genetic perspective?
Gosh, that's,
Can we make this system even more efficient by combining the attributes of semi-confinement and some aspect of reducing maintenance requirements on the genetic side?
great question. I think potential is there. The maintenance, looking at the genetic aspect of maintenance, I think it falls along this scale a little bit too, is that is a complex trait, and so what are some of the individual component things that affect the maintenance of the cow? And so we know from somewhat limited studies that there are differences, genetic differences in some of those components that might impact maintenance. And so it looks like there's potential to select for lower maintenance. And the most effective way to do that might be to better describe some of those component traits that affect maintenance. But it looks like the potential's there to do that.
Any other tough questions for Andy? All right, let's give it.
Thank you.